Hello everyone, my name is Mathieu Rouard. I work for the Alliance of Bioversity International and CIAT, where I lead a team of bioinformaticians that are linked to the International Gene Bank on Banana. So even if I cannot be with you today, and sorry for that, I'm still very happy to have a chance to talk about the management of genotyping data in gene banks. I'm sure my colleagues Marie-Noël, Carolina and Monica already highlighted what we mean by digital sequence information in gene banks and how important they become over the past years for conservation and use of genetic resources. But it's also very important to remind and stress that handling DSI can be also very demanding. Each time you take a plant to conduct an experiment for sequencing, whatever the machine you use, what you get in return are usually terabytes of data that are challenging to manage unless you have access to appropriate storage, analytical tools, and computing resources, as well as access to experienced bioinformaticians to help you to deal with the data. But even if you can get rid of these constraints and difficulties, what you will get at the end are genotyping data files that are very big files that are very difficult to manipulate if you don't have the proper skills in bioinformatics to do that. So what you see on the screen here is a type of file, genotyping data file, that is called a VCF for variant call format, where you have information about your genetic markers for all your samples. And you can have in this file millions of lines. So obviously that is very difficult to have a global overview of your dataset, to explore it, and also to change it if you need. Basically what we need is, if we work in gene banks, is to collect this genotyping data, to send it to a system that would allow to store and share the data for the user of the gene bank. That can be the gene bank itself, but also multiple labs in the world, breeders, farmers, and so on. And to be able to check the data, to adapt the data to our own needs, and to export them to do subsequent analysis. To check, for instance, the diversity of your sampling, to do population genetics, to look at the genes that are close to your markers, to look at QTLs, or to look at diversity of chromosomes. So you have many examples of analysis that you can do with that. But those data in itself are not enough. What you need is also to be able to connect with metadata, like the passport data that you can find in Genesis. And this type of tool actually exists, and there is one that is called genebanks. So we have been collaborating on this tool for many years now with CIAT and IRD. And what it does, it stores large amount of genotyping data. So billions of markers can be stored in the database. And then it provides an online graphical interface to share the data with the users, to explore and visualize the data, to filter the data, to browse very specific markers and genotypes, and then to export those subsets in various formats and external tools. So genebanks is open source, it's free to use, and it can be set up in any type of platforms. So let's give an example of what we mean by filtering. So I want to see what is the marker distribution along chromosomes. So I can do that. I want to know how many markers I have with a very good quality score, but less than 50% of missing data, if I want to do diversity analysis, for instance. Other people would like to have a list of markers that are very discriminating between two groups of population, between wild, between cultivated accession, or between genotypes that have been used for stress, and you want to compare with the control. So you can see exactly what are those markers in the application. Or you may want to export a very specific region on a chromosome because you know that are genes of interest, and you want to see what are the markers and visualize that in a specific tool for genetics. Or you want to know, because you know one marker of interest, you want to know what are the genes that are nearby, or if your marker is in one gene, and does it affect the structure of the gene. So everything can be actually done in genebanks. And we have applied the system to the international gene banks for banana. And what you see on the screen is an article that we published recently where we described how we can take advantage of the system. So you can see it at the bottom of the screen. This is the type of interface that you can manipulate with GIGWA. And we compiled a number of studies that have been done for GWAS, for analysis of chromosome structure, genomic selection. So everything was put in the system, and now people can access that. It's free to access, it's open access, there is no restriction. So we are not the only one to use it. It's also being used by ICARDA, by the gene bank at ICARDA. It's being discussed for other gene banks in the CGIAR at the moment. It's also being used by national research organizations in France, CIAT and IRD, because they are very active in the development of the tool, but also in some lab at USDA. And it's also being adopted by some breeding system, because there is also the same type of needs when you want to deal with genomic selection, for instance, in breeding programs. I want to conclude this presentation by talking about capacity building, because this is really at the core of the question about DSI. How to facilitate access to genetic data if you don't have skill in bioinformatics, how to facilitate online access for many organizations, many labs. So I think GIGWA can allow that, and we have been involved in a master degree over the past years at the University of Montpellier, where we teach students how to take advantage of large genome sequence data when you want to study the diversity of important crops like rice, banana, sugarcane, and others. So for that, we took the example of the 3,000 genome of rice, and we show to students how they can export data for those 3,000 genomes that are difficult to manipulate. And this course actually raised interest in other universities around the globe, and there will be some type of adaptation courses in Senegal and Portugal this year that is led by the AGAP Institute. So with that, I would like to thank you for your attention. So if you have any question, don't hesitate to send me a message. My address is at the bottom of the slide. I'm sure also my colleagues can take a few questions, and I give example of article about the system I have described in this presentation if you want to look for more information. So thank you very much again.